Good morning to the ones participating from America, and good afternoon to or good evening to the participants from Europe, Asia, Africa, and Oceania. I want to thank everyone who honors us with their presence, and as well as anyone who has helped in spreading the word for the webinar. I will be brief in my introduction because a demonstration is threatening to pass through the road outside my office any minute now. So there will be a lot of noise. My name is Georgios Pappas. I'm a physician with an interest on zoonosis. I am currently chairing the zoonosis working group of the International Society of Antimicrobial Chemotherapy. The society was founded in 1961, is a federation of affiliated member societies, and aims to increase the knowledge of antimicrobial chemotherapy and combat antibiotic resistance around the world. The society currently has a worldwide membership composed of 99 national or regional societies, which in turn have more than 60,000 individual members. It hosts the International Congress of Antimicrobial Chemotherapy every second year. The next Congress will be November 2024 in Istanbul, and the program has just been uploaded. You can go through it in the relevant site, in the relevant website. So, without further delays, why do we care about avian influenza and why we are hosting this webinar? We have been discussing about H5N1 since 1996, 1997. It seems like a threat never materializing for humans, at least. What has changed in the recent months, in the 2 recent years, in 3 recent years? Many important aspects: its expansion, both geographically and in terms of susceptible species and severity, its steps towards mammalian adaptation, and this mammalian adaptation may herald the risk for human adaptation, and its potential in such a case to induce a pandemic where no one theoretically will be immune to such a strain. So we aimed at gathering the leading scientists worldwide in its aspect of H5N1, and we are really honored and happy that they have responded immediately, and we have them here with us. So the first speaker will be Dr. Isabella Monne. She is a veterinarian and currently head of the Viral Genomics and Transcriptomics Lab at the Istituto Zooprofilattico Sperimentale della Venezia. The lab hosts the European Union Food Agriculture Organization, World Organization for Animal Health Reference Lab for Avian Influenza and Newcastle disease. Dr. Monne, since 2016, is the World Organization of Animal Health expert for avian influenza and Newcastle disease, and her research focuses on the use of sequencing technologies and bioinformatics tools to investigate the evolution of RNA viruses, in particular the ones affecting avian species. So, Dr. Monne, you can start, and thank you once more for being here with us.
Thank you very much, Georgios, for the kind invitation and the kind introduction. I start sharing my screen. Okay, in the next 30 minutes, I will try to summarize the unprecedented challenges caused by H5N1 viruses in the most recent years. And I will try to do it focusing on the animal perspective, as you can understand from the title. Well, migration is a vital survival strategy for any individual in this planet. And it is in particular for wild birds. Every year they do move across the continents, within the continents, looking for food or to breed. Unfortunately, their migration are happening in a world that is constantly changing. And there have been an increasing number of risks for their extraordinary journey. Since 2005, there's a new one, which is represented by highly pathogenic avian influenza viruses of the goose Guangdong lineage, which, have been causing unprecedented concern and, let's say, forcing us to review many aspects about influenza. The first one is really related to the role of wild water birds. I do remember when I was at the university studying the epidemiology of the disease, and wild water birds were just considered the natural carriers of low pathogenic avian influenza viruses. But the goose Guangdong lineage has forced us to review this situation and have a new thinking that consider wild water birds as vectors and victims of highly pathogenic avian influenza viruses. Well, so the normal scenario where highly pathogenic sporadically emerge in poultry but stay as an issue for poultry has been confirmed multiple times. This slide is summarizing all the highly pathogenic avian influenza viruses of both the H5 and H7 subtype that have been emerging in the last century. Well, all of them have been characterized by an association with the poultry production. And even starting from 2021, when, the 21st century has been characterized by an impressive increase in the number of highly pathogenic viruses emerging in association. We have an increase in the global poultry production. They have been maintained main as an issue for poultry and successfully eradicated in most of the cases. This was not the case for the H5N1 virus that emerged back in 1996. The goose Guangdong lineage was totally changing the scenario. And starting from 2005, we are in front of this strange situation. We are living a vicious circle when there's continuous spillback and spillover between domestic and wild birds. What is unprecedented is the role of wild birds as source of virus. Recently, this role has become more and more evident considering the different transcontinental epidemic wave. And I want to use this Bayesian analysis to give an objective demonstration about this increasing role as source for wild birds. You have just to check the color. Brown is for domestic birds and the blue one is for wild birds. If you look at the different clades that have been able to make a transcontinental transportation of the virus, starting from 2005 with the clade 2.22 and moving up to the 2.3.4b in the upper side of the slide, you can see that the blue color at the trunk of the trees are becoming more and more evident. This means that there's an objective evidence about the role of wild birds as new source for viruses around the globe. And this is happening in parallel with a broader host range, an expanding list of birds found infected by the virus. This slide summarizes the number of species that in most recent years have been affected across the different avian orders. It's impressive to see how many species and how many regions have been affected with the appearance of exotic is like endemic in humans, an exotic situation even in region never affected before by exotic events in wild birds, such as Europe, the Americas. So the high number of wild bird species affected and moving, the high environmental contamination has resulted in the situation that you can see summarized in this slide, with outbreaks recorded everywhere, including South America, never affected before by descendants of the goose Guangdong lineage. This comes with many issues. Unprecedented was, as an example, the number of mass mortality events that have been occurring in the last 3 years. Across the continents, there have been multiple species affected. The situation is like the virus arriving in a new scenario is able to occupy new ecological niches and in fact new species, causing in many cases unprecedented number of birds dying because of the infection. And what we know so far is just a drop, most probably, of the real scenario, because these are wildlife and it's difficult to monitor the situation. What is clear is that there have been already species which are vulnerable or endangered that have been affected by the virus with great concern for their future. But there's other species that may assist to a change in their status. I'm referring in particular to the mass mortality events that have been involved with species which have, let's say, a low production of progenies, the long life animals such as some seabirds. What is going to happen in the future? How many other species are going to be threatened because of highly pathogenic virus? Well, the situation seems concerning also thinking about the areas that have been affected. These only places for biodiversity, such as Galapagos, and now, since last month, even Antarctica region, with all these species that are endemic just to this region that are now threatened by the virus. And it's not just an issue for wildlife, it's clearly also, let's say, a routine issue for poultry. If you look at the war reports that are published every 3 weeks, and you look at the last 2 years reports, it's impressive to see how many thousands of animals have been dying or culled to control the disease across the world in the recent year. This is extremely concerning for the animal health perspective, first of all, because even them are just farm animals, they are extremely valuable as any other animal, and because of the social and economical consequences. I reported here a picture from the EFSA journal, which is covering the European situation. Just to give you an example about how dramatic it is, the situation. We had, in the last 3 years, 3 consecutive epidemic waves. This was absolutely unprecedented. And we are now actually entering the 4th consecutive one, because now the number of cases, at least in Europe, but even in the Americas, as far as I know, they are increasing once again. And what is unprecedented is also the persistence of the virus. I mean, we were used to have a pause during the summer season, at least for the circulation in wild birds. But now the situation is very different. If you look at this graph and you check for 2022 summer season and 2023 summer season, well, you can see a rise in the number of wild birds affected. And this is related with the emergence of a new genotype that is very well adapted to infect seabirds. And in particular, there have been dramatic events happening in colony breeding seabirds. This virus has something making it so fit for purpose, I mean, which is the secret? We do not know, but the emerging genotypes and the ability to generate new genotype maybe it's a successful key. This tangogram describes how the different clades, descendant of the goose guangdong lineage, have been evolving across the different phylogeny, from the hemagglutinin to the MS gene. If you look at the violet color as an example, which is the one for the first clade able to make a transcontinental spread, called 2.2, you can see that the violet color, however, is maintained with clustering across the phylogeny. If you look at the red color for the 2.3.4.4b clade, you can see that the line and the genes are spread across multiple parts of the phylogeny. What does it mean? It means that a lot of reassortment has been happening with this virus. And this is well resembled if you look at the number of genotypes that have been emerging in distinct continents. I mean, I can easily describe the situation in Europe, the area where I come from. We had 73 distinct genotypes in the 3 consecutive epidemic wave, and a lot of diversity. All these colors you can see here refer to genes that the virus has been acquiring from multiple low pathogenic avian influenza viruses circulating in wild birds. And only few genes have been maintained together with the hemagglutinin of the 2.3.4b clade. This is the case of the M gene, which has been maintained across the multiple genotypes that have been emerging in the distinct epidemic waves. And starting from 2021, we can see that also the neuraminidase has been maintained. And this is particularly interesting if we look at the data from other continents, such as North America. There was this recent publication explaining the genotypes that have been identified in North America. It's interesting, it's very interesting to see that even in North America, the same 3 genes that were maintained in Europe are the same that have been maintained also in the North American region, suggesting that these 3 viruses, these 3 genes should have a sort of selective advantage. M gene is indeed involved in the virion morphology and is crucial for the balance between HA and A on the surface. So it could be that this may have been driving their maintenance. But maintenance of Eurasian gene in the America is also something unprecedented, because we were used to have viruses sampled for birds of the Americas, very distinguished from the Eurasian one. This phylogenetic tree is explaining that the genetic structure for all the different subtypes is generally based upon avian geography. But with the clade 2.3.4.4b, we are assisting to a globalization of Eurasian genes that are now mixing in the American setting. And this globalization has been also happening in a, let's say, unprecedented way. There's new trajectory that the virus has been using. In the past incursion of descendant of the goose guandon lineage, we assisted to the movement of the virus mainly from Asia toward the western side. And even the time that the virus was able to spread in North America, it was through the Bering Strait, from Asia to North America. We implemented an extended analysis in order to understand where the current 2.3.4b virus has been moving from. And it is actually interesting to see that regions southern to the Mediterranean Sea has been most probably the source for this virus. Since then, the virus reached Central Asia, and from there, as happening all the other time earlier, the virus was able to move in multiple other regions. But the other jump that was unexpected is the one toward the North America. Europe has been the donor this time for North America, even if another source also happened later. But it was the first time that Europe was, distributing viruses abroad. And this situation has been mostly and probably the consequence of an unprecedented, let's say, spread northern and western of the virus in Europe. You can see here in red the most recent event involving also Norway and Iceland, and most probably from there, with the birds infected there, the virus moved to North America. There's already another new thinking possible, and it is related not only to the evolution of this virus in wild birds, but even the evolution of this virus in poultry. Back in 2017, for the first time, H5N1 viruses of the zoonotic G1 lineage have been, let's say, moving toward West Africa. Now H5N1 subtype is endemic, and this maintenance of the virus together with the persistent circulation of the H5 virus has favored the emergence of new resortant strain, both for the internal genes, but also for the surface glycoprotein neuraminidase. This is concerning if we think that H5N1 viruses have already been working as, let's say, a component for the gene pool of viruses, avian influenza viruses with an increased zoonotic potential. And that's another important aspect that is emerging. All these different genotypes that have been emerging and spreading has been also posing new questions about the pathogenicity of the virus. We are used to think about highly pathogenic avian influenza viruses as causing disease. But we know very well since 2005 that wild and domestic waterfowl may be infected without presenting any kind of clinical disease. But it's unprecedented to find silent infection in galliforms. I try to share the experience we had in Italy, but it's common to many countries. We had a very serious epidemic back in 2021, 2022, and during active surveillance, so what we did was actually to check broiler anytime they had to move. And so what we found was 15 broiler farms infected by the highly pathogenic H5N1 virus without presenting clinical signs. And even an in-depth investigation later on in one of the farms confirmed that the mortality in the next 72 hours was within the average. And the unique way to identify the virus was to check the few dead birds that were present at the farm. This is quite concerning if we think about the other basic principle that has been, let's say, changing since the emergence of the goose-Guangdong lineage. Is not anymore sporadic, in particular with the most recent 2.3.4.4b clade, to see infection in mammals. There have been an impressive increase in the number of species affected. And unfortunately, also mass mortality events have been reported in mammals. I'm referring to the cases occurring in the Americas. And right now in Argentina in particular, thousands of sea lions and elephant seals, both puppies and adults are dying. And we are just recognizing the ones that are, let's say, moved to the land, but the number may be even greater than the one we know. And it's impressive even more to look at the data from the kept mammals. I'm referring in particular to the fur farms, which have been affected both in Spain and in Finland. Finland now has reported 42 affected farms. This is impressive if you think also about the number of animals that are kept inside for their furs, from minks to Arctic foxes, raccoon. They are sometimes many animals inside kept together that can, let's say, provide a sort of experimental trial in the field to make the evolution of the virus possible. The other concerning aspect is related with an event that occurred once again last summer, 2023, in cats in Poland. There was 35 cats, 1 caracal and 1 dog affected by the virus, all of them affected by a unique genetic cluster, so a unique genotype causing infection of all these animals. And the question clearly for all these events summarized here briefly is, which is the source of infection for all of them? Well, in most of the cases, the link with the wild bird infection is, let's say, the most likely one. But in some cases, there's an uncertainty about the origin of the outbreaks. And I'm referring in particular to the situation in cats. Oops. Our Polish colleagues did implement an in-depth epidemiological investigation in order to understand which was the source for the infection of cats. Unfortunately, it was not possible to get to a final conclusion, but based on the results of their investigation, raw poultry meat was demonstrated to be the prevalent feeding method. And there was also identification of a chicken meat sample by another group of colleagues in Poland. And the genotype identified in that the chicken meat sample was exactly the same identifying cats. So there's the chance that the virus may enter using food, the pets, and this has been happening even in South Korea, where H5N1 virus was confirmed contaminating cat food and affecting them through cat food shelter cats. But we are inside a puzzle that is even more complex than this one. And there could be trajectory of transmission even different than this one. If I can, I would like to share this experience we had in Italy last April 2023. We identify a backyard farm infected by H5N1 virus. And checking the sequences, we identify in the PB2 gene a mutation called 271A. And this mutation is considered a markers for mammalian adaptation. It was unprecedented for us to find this mutation in an avian species. And for this reason, with the endorsement of Ministry of Health, we implemented an additional surveillance in mammals present at the farm. The results were quite surprising because we found 5 dogs and 1 cat infected, seropositive for the H5N1 virus, but with high titer confirmed by H5N1 test and ELISA also. So they were all healthy. And the question is then now, who infected who? The virus identified in that avian species was presenting a mutation that may indicate that we had some bridge species that we were not able to identify. And this gap in knowledge is quite concerning. So summarizing, we are in front of a panzootic event inside a panzootic event, because there are multiple genotypes that have been emerging, and every new genotype is posing new questions about which is its phenotype, how they are behaving. It's like the situation we have been facing with SARS-CoV-2, any new variant posing question, but in that case, there was a sort of global effort in order to understand what was happening, pathotyping, understanding the efficacy of vaccine. For avian influenza, we need a lot of improvement. The pillars for improvement stay in one hand, but there's a lot of actions that are needed. First of all, we need to go beyond the tip of the iceberg. We need to understand much more through surveillance and through characterization of what we are finding. What we are understanding right now and what we know right now is what is mainly stipulated by law. But we need to know much more about mammals and circulation of the virus in there. We need to know much more about wild birds. Indeed, we were used to do surveillance in wild birds to inform risk for poultry. But now we need surveillance in wild birds also to understand which is the impact on wildlife conservation. Because the risk in case we lost diversity is imperative for all of us. We need also to know more about these events that are occurring in wild birds. How many animals are dying? We need to do monitoring about the mortality, because understanding how many animals are dying means understanding which conservation action we can make possible. Even because if we have a species that is reducing its number, the virus itself can find other way to evolve and jump to new species, occupy new ecological niches, and it's clearly a risk. We need to identify the virus, how persistent it is in the environment, and find sustainable solutions to limit the environmental contamination. There's then another aspect related with connection. This pillar need absolutely to find a solution for real-time sharing of information. In the animal side, it's not that easy maybe than in the human one. We have multiple barriers. The barriers are for sure economical, but there's also motivational and political barriers. We need to remember that behind the animal production, there's economy. And we need, to solve issues related with lack of trust. Sometimes people are scared to share because they are worried about how the data are going to be used. And we need to build a trust. There's then the need to have a multi-sectoral approach. And this needs to be the new normal. if you go and analyze all the determinants of the disease across the different area, you can see that both in the private and even in the public system, there's a lot of sectors that may have an interest on this disease. And we really need influencers to involve them. First of all, because we need funds to address the major question we have right now. And we also need minds on this issue. We need resources, we need, new approaches to solve the problem. We need to empower even for communication because, as an example, general public is crucial to report mortality events in wild birds, but it's also crucial in terms of their protection. We do not have to cause alarm. We need to inform them about the risk associated with these viruses. We need to empower clearly research purposes, both for vaccine development, diagnostic tools, and we need now absolutely to work much more on prevention. I do remember when I was working as a veterinarian in a clinic, anytime a dog passed away, there was this statement, we did everything we could, which is the same that in the human sector. With the animal protection, what we can comment is we did the best we could, and we are actually working as crazy in doing a lot, but we are still assisting to millions of birds that are killed because of the control measure. In the short term, we need to find new solutions for prevention. As an example, in case there's an increased risk of influenza introduction in an area that is densely populated with many poultry farms, we need to have resources to support a temporary ban on restocking farms, anticipating the slaughtering of animals. So all the way in order to reduce the intensity of farming. We need also to optimize biosecurity measures, but enter the food chain, and also in the natural settings, because it's extremely important for both the protection of wildlife and also human beings. In the short term, we need also to remember that in our toolbox, we do have vaccination. And in high-risk transmission area and in poultry species which are highly susceptible to the disease, we need to think about using it. In the long term, we will need better vaccine, absolutely, because we need something that is cheap. We need something that can be deployed for mass application. But we need also to change the way poultry are reared and sold. We need to think about animal and human health consideration driving our decision, and a bit less the market. And we need also to consider a priority protection of wild habitats. And I want to thank all my group, which have been intensively working in the last 4 years, and all the European Union projects which have been funding some of the data I've presented here. Thank you very much for your attention. Over.
Thank you, Dr. Monne, because you've made it extremely clear for everybody why something is changing and why you should be worried. There are some questions that will be answered in the hall after the 3 speakers. So we will proceed with our next speaker, who is Dr. Thomas Peacock. Dr. Peacock is a fellow at the Pirbright Institute and an honorary lecturer at Imperial College London. He is working on interspecies transmission and adaptation of influenza viruses. Thomas studied for his PhD at the Pirbright Institute, working on the receptor binding and antigenicity of H5N1 Avian Influenza viruses. You've been hearing for H5N1 in China in recent weeks. This was followed by a short postdoc at UCL, researching innate immunity and HIV. And then Thomas did the second postdoc at Imperial College London, initially working on the role of influenza host factors on interspecies transmission, and then later working on determinants of entry and transmissibility of SARS-2. Additionally, Thomas has been one of the voices of a scientific reason in the public sphere during the pandemic. And this is also invaluable because we need to communicate science, and he has excelled also on this. So, Dr. Peacock.
Thank you. Thank you for a very kind introduction, and for Isabella's fantastic talk before this, which covers a lot of the background, which makes my life a lot easier. So I'm going to go into a little bit more in depth about. Can you hear me? Okay, by the way, and see the slides moving.
Yes.
Okay, fantastic. So I'm going to go into a little bit more in depth about H5N1 in mammals and what sort of evidence we're seeing for mammalian adaptation and how this might impact on the pandemic potential of H5N1 viruses. So kind of starting off, what does it actually take to cause a pandemic if you're an influenza virus? And it turns out there's quite a lot of barriers that you have to overcome. Particularly avian influenza viruses are really not very well adapted for infecting and transmitting between humans. But some of these barriers are quite easy for viruses to overcome, but other barriers seem to be more difficult. So I'm going to cover what some of these barriers are and what we're seeing out there in H5N1. So it's worth saying there's kind of two different types of infections that we worry about from a public health point of view, but they are intrinsically linked. And these are zoonotic infections, so being directly infected by animal viruses. But when a zoonotic infection happens, there is a small chance that virus might be able to adapt, or even it is already adapted, to then transmit from person to person. And if this is happening efficiently, this then causes a pandemic. So zoonotic infections seem to be, well, for influenza viruses, fairly common, whereas pandemics happen every few decades. So it's worth saying this is not an exhaustive list of the sorts of things that influenza viruses need to do to go pandemic, but these are four that I think are quite important and that we understand fairly well. So one that's very close to my heart is this idea that avian influenza viruses can't actually make copies of their genomes very well when they get into human cells. They're severely restricted. We understand a little bit about the molecular basis of this, but what has to happen quite quickly is the virus needs to get mutations that allow it to overcome this barrier. So one of them that you'll see me talk about a little bit later is a mutation called PB2 E627K. And this is quite a common one, and this is a mutation that's seen in a lot of pandemic viruses as well. As well as this, as they replicate in human cells, there are certain intrinsic restriction factors. So these are proteins that block infection or slow it down at least. And there are a number of these, but two that I'm going to talk about a little bit today are called MXA and butyrophilin 3A3. And both of these, the human versions of the proteins, generally block avian viruses from being able to replicate efficiently. However, we know what types of mutations allow overcoming of this, and they're fairly easy for viruses to get. We see it happen routinely. So another barrier is that in the human airway upper respiratory tract, we have the wrong types of receptor that avian flu likes to bind. So flu binds sugars that are called sialic acids, and these sialic acids are found at the ends of sugar chains. And the type of linkage that they're bound to the next sugar is a determinant of this. So whereas birds in bird guts, there is one type of sugar, is very abundant, and avian influenza viruses learn to use this very well. The version that's found in the human upper respiratory tract is a different kind, and therefore the virus has to learn to go from this avian-like receptor to a human-like receptor. And we understand a little bit about how H5N1s can do this, but it doesn't seem to be very easy. It's not always straightforward. Finally, there is avian influenza viruses are not thought to transmit, particularly H5N1s, are not thought to transmit efficiently by the airborne route. So it's instead thought, although this is maybe slightly controversial, that H5N1 viruses in particular, and particularly in waterfowl, are spread more by the fecal-oral route or through environmental contamination, potentially. Whereas human influenza viruses definitely transmit by the airborne route or by fomites. But both of these routes require a virus that's really quite stable. So what you see when viruses go pandemic is they start off as an unstable avian influenza virus, and then they become a stable human influenza virus. So one final thing, which Isabella covered in really nicely, thinking about general circulation of H5N1s, is reassortment. So this is the idea that two influenza viruses can infect the same cell or the same host in the same cell at the same time and exchange gene segments. So we know that the three last influenza pandemics all emerged through reassortment events, and these, some more complicated, some more simple, were at least a mixture between avian viruses and human viruses. So someone or an animal that was infected, or a human with both an avian and a human virus at the same time, and the virus kind of got the best of both worlds. Because of influenza's segmented genome, it could kind of mix and match exactly what it wants. And when we think about mixing hosts for influenza, we think about pigs in particular, because they can be infected with both avian and human influenza viruses. But more recently, people have been thinking a little bit about mink, which I'll get into a little bit more in. So when we, as Isabella said, in some ways, H5N1 is the worst it's ever been. It's on the most continents, it's killing the most wildlife, it's infecting the most mammals. But there is one thing that hasn't happened so much recently that has been an issue in the past. So between 2004 and 2015, there were a lot of H5 human cases. And these had a very high case fatality rate, although this is almost certainly an overestimation based on only very sick people were being tested. And at the time, there was no recorded transmission, though there has been recorded transmission of other avian influenza viruses more recently than that, or putative transmission. So in some ways, we haven't hit that peak. We haven't seen the amount of human cases, despite the virus being much more widespread. Some of this may be to do with biosecurity. Some of this may be to do with changes in the virus itself. So we are seeing sporadic human cases, but quite often this is being picked up, with a few exceptions, when people have quite mild illness. So very few people have been tested positive, but even fewer have died or even had severe infection. And this might be partly, again, an ascertainment issue. So WHO had recommended countries undertake asymptomatic surveillance of poultry workers who'd been exposed to the virus. So, post-pandemic, everyone is now quite happy sticking a swab up their nose and sending it somewhere. And these programs have taken advantage of that. And yeah, a lot of these infections, it's not even clear if these people are actually infected, although they may be testing positive for viral RNA. It's possible they may have been, been breathing in dust that contains non-infectious virus, for example. It's worth saying as well, these human H5N1s of the 234.4Bs, there's not been any evidence of adaptation, which is not the case with mammals, as I will cover now. So when we look at non-human mammals that have been found to be infected, Isabella gave a really good breakdown of the wide variety that have been infected. And here's a word cloud on the right here that kind of covers some of that. Lots of foxes, but also skunk, mink, seals, sea lions, domestic cats. There are a number of other species that there were too few, but things like dolphins as well have been found. So the largest group outside fur-farmed animals is scavengers, so particularly foxes, things like otters and skunks as well, which would probably eat a dead bird, as well as this, we've seen a lot of aquatic mammals, particularly sea lions, but also seals and dolphins, a number of companion animals, particularly cats. And then the last thing is fur farm carnivores, so particularly mink, but also foxes and raccoon dogs, which I'll talk a little bit more about all these in more detail. So it's not necessarily surprising to find H5N1 in scavengers because, we know, here's a picture of a fox carrying a dead bird. You can absolutely understand that a fox is going to be very highly contaminated by virus if it's eating or picking up dead animals, dead birds that have died of H5N1. So we see this particularly in foxes, but also some mustelids. What we do see is quite a lot of neurotropism in these animals. So this is quite a nasty set of, things like tremors and spasms and things like that. This is also seen in chickens, it's worth saying, for some highly pathogenic avian influenza virus strains. But generally, although it's very difficult to tell because these are wildlife, we don't think we see much evidence of mammal to mammal transmission. We think generally these are probably coming directly from dead birds that are being eaten. However, that's not the case for cats. So as Isabella said, there was this very large cluster of cats infected in Poland. Many of these cats were house cats. It's still not been quite confirmed where this came from, but it's thought it may have come from contaminated raw meat. So there is a large trend at the moment of feeding cats raw chicken, basically, because it's I think the cats, if you have a fussy cat, they tend to prefer that. And it's thought there was at least some testing of some raw meat from one of the houses, and it tested positive for H5N1. A similar outbreak also was found in cat shelters in South Korea. Again, it's not quite clear if there was cat-to-cat transmission or they were all eating the same meat. They were obviously, all the cats were in the same place, although it did happen at multiple shelters. And actually that very quickly, the Korean outbreak was linked to a certain brand of raw poultry meat that was being fed to the cats. And what was interesting, particularly about the outbreak in Poland, is in the PB2 gene, as I said earlier, we think of one of these, PB2 is where some of the mammalian adaptations come. There was a pair of quite unusual mammalian adaptations, E67K, which I mentioned earlier, and another mutation. So this is a little assay that we did where we take a polymerase of flu in a reporter assay, and we can measure how much activity it has in human cells. So if we take a human H5N1, we see it's got very good activity. If we take a purely avian virus, we see it's got no activity, and then these two mutations boost the activity quite a lot. So there's still a lot of questions to be answered about what happened in Poland. Unfortunately, the time has kind of gone by and we still don't have a lot of those answers, which is a little bit disappointing, particularly as there was some indication that the meat that was being fed to the chickens was potentially human meat that was, for human use as well. And it's unclear why these viruses had these mammalian adaptations, which may link back to some of Isabella's points about sometimes chickens do appear with mammalian adaptations, and it's not clear if there's been a mammal around in the past that's been infected. So finally, of particular concern more recently have been the outbreaks on fur farms, particularly in mink. So as Isabella said, these have happened in Spain and Finland. So one real worry about mink in particular is when we want to see if a human or an avian influenza virus transmits well in humans, we can't really stick it up a human's nose. So what we use instead are ferrets. Ferrets have a number of advantages. They sneeze, just like humans do when they get flu. But as well as this, they seem to have a very similar receptor distribution as humans due to upregulation of certain enzymes involved in the receptors. No other mammals really have this, as far as we know. However, mink are quite closely related to ferrets, so we do worry that mink and ferrets could do the same thing in that they could drive human-like adaptations in an avian influenza virus. And when we think about how mink farms are set up, as you can see on this picture on the right, these mink are very close together, they're at very high concentration, and they have little gaps between their cages. And I'm going to show you a picture in a minute of how ferret experiments are set up, and it's very similar to this, but obviously with extremely low biosafety compared to a ferret experiment in the lab. So there is a little bit of evidence from these mink outbreaks that there may be some mammal-to-mammal transmission. So sometimes these viruses in Spain and in one of the outbreaks in Finland share some fairly unique mammalian adaptations, which might be suggestive of mammal-to-mammal transmission. And then here are the mutations that we saw in Finland and Spain. So 271A, as Isabella was saying earlier, in the outbreak in Italy was also found in the mink in Spain, whereas 627K was found in Finland. And you can see, you can't see a purple bar there because there's no activity in human cells. But when you put either of these mutations in, you can see that the virus can replicate its genome. And finally, as Isabella touched on again, there's been huge outbreaks of sea lions in South America, so Chile, Peru, and most recently Argentina. And again, we can look at the genomes of these and we can see quite an unusual combination of PB2 adaptations that have been found in several of the sea lions in both Peru and Chile. This might again suggest mammal-to-mammal transmission and adaptation. What's slightly concerning is the human case in Chile also had this pair of mutations. It's unclear exactly where they were infected from, though they had walked on a beach where there had been both dead sea lions and dead sea birds. So it's possible that that was a sea lion to human infection, potentially. The mutations would be consistent with that. So finally, I mentioned, or not finally, but I mentioned earlier about these restriction factors. And we had a look if the current circulating viruses, so this group up here, these are genotypes from that we'd found in the UK, were sensitive or resistant. So what you can see here is if the blue or the red bar are at the end, they're resistant. So you can see human and zoonotic influenza viruses are generally resistant. We had some older H5N1s that were not very zoonotic, and we could see that they're mostly sensitive or partially sensitive. But when we look at recent viruses, they are nearly all resistant to butyrophilin, and they're partially resistant to MXA. So we think these viruses probably won't have much issue infecting human cells, at least from these restriction factors. So we think that there is probably evidence for some mammal-to-mammal transmission. This evidence is, there's hopefully epidemiological investigations ongoing still that can say this, whether this happened for sure or not. But particularly in the sea lions and the farm mink, there are mutational signatures to suggest some degree of mammal-to-mammal transmission. And this would be a first for H5N1. And finally, this is what ferret setups look like in the lab, where you basically put a ferret, you infect it, and then you have a perforated gap between that and the next ferret. You may notice that the mink farms look extremely similar to that. So a number of studies have looked at H5N1s in ferrets to see if they're transmitting. There's a bit of study-to-study variation. There's been a little bit of transmission reported, but this is inconsistent and it seems to be fairly inefficient. Ferret experiments nearly always involve quite low numbers and could be quite stochastic as well. So I would say generally the virus doesn't, or depending on the study, doesn't appear to be transmitting well between ferrets. So let's kind of summarize this. Where are we now? What are we seeing out there and what are we not seeing out there? So we're certainly seeing polymerase mutations. These viruses seem to pick it up. Every time they enter a mammal, they'll pick it up, or on almost every time. We also think that these viruses are very resistant to restriction factors. However, both of these things seem to be very easy for H5N1s to evolve. When we look at the things that are more difficult to evolve, so we haven't seen any evidence of reassortment with human viruses, although there's been a lot of reassortment with other avian influenza viruses. Even in all these mammals, in the mink viruses, there's no evidence of human receptor binding, which would be a real red flag if we were seeing that. And other subtypes, when they infect humans, other avian influenza virus subtypes, sometimes within a single human, we see adaptation to some human receptor binding. As well as this, we don't think, as far as we know, there's any very good evidence for airborne stability. So maybe that's, not as bad as it could be. We're certainly seeing some things, but not others. So just kind of on a slight tangent, there are other subtypes of influenza, and although everyone's looking very closely at H5N1, I personally think there's a very good chance the next influenza pandemic is not something like H5N1, and it's something a little bit more boring and a little bit more familiar. So, for example, Eurasian avian H1N1s. In fact, we've seen a cryptic H1N2 case in the UK in the last couple of weeks, which doesn't appear to have come directly from a pig, so maybe a short human transmission chain, as well as this H3N8 is a virus that's becoming quite widespread in China. That is the virus I mentioned, which does get human adaptations when it, human receptor binding when it enters human population. This also has internal genes from an H5N1, which are the same internal genes as the H5N1 virus that you'll all remember from a decade or so ago in China. So this is really quite a nasty virus and really one to keep an eye on. So with that, I'd just like to thank the folks who generated the data and that I've been talking about today, and of course all the sequencers and sequence submitters, everyone who shares their sequences, particularly in a timely manner, which makes all the virological downstream analysis much easier and really helps. Cool. Thank you very much. That is my talk.
Thank you, Dr. Peacock, for the presentation, but also for the optimism of the current situation. Now, as I have said, the questions will be answered in the end. So our final speaker is Dr. Charles Todd Davis. He is the acting branch chief of the Virology Surveillance and Diagnosis branch of the Influenza Division of the CDC. He leads the Influenza Division's efforts to improve global influenza virus control, prevention, pandemic preparedness, and response. The branch conducts comprehensive virus surveillance to analyze antigenic, phenotypic, and genetic characteristics of viruses circulating in humans and animals. And the data is used to guide influenza vaccine strain selection, assess virus sensitivity to therapeutics, develop or assess diagnostic tests, and generate candidate vaccine viruses for both seasonal and pre-pandemic influenza, which has already been a question multiple times in our Q&A session. So, Dr. Davis, we will be happy to hear you.
Thanks very much to Isabella and Thomas for the fantastic presentations. It makes my job a bit easier. I will try and focus on the public health response and preparation for what may inevitably be another pandemic. I'll focus on H5, but as Thomas concluded his presentation, of course, there's quite a lot of other zoonotic influenza viruses that have been detected in humans and quite a few subtypes that are circulating in animals as well. So I'll touch on a little bit of this as I go, but just to begin, from a public health perspective, we like to think of pandemic preparedness as very much being part of our seasonal influenza virus preparedness. And I'll go into some of the details of why we think that's important. But as everyone is keenly aware, while we do follow the seasonality of human influenza viruses, we also follow trends in zoonotic influenza viruses. And as Isabella really pointed out nicely, there's been quite an interesting trajectory as these viruses have evolved over many, many years in different types of hosts, and in particularly avian hosts. In the US, at least domestically, we do focus our surveillance efforts by coordinating activities with public health laboratories across US states. And so we are constantly receiving specimens from clinical laboratories, hospitals, as well as state public health departments that submit samples to the Center for Disease Control for characterization. So on average per year, we receive about 200,000 influenza positive specimens through our state and local public health departments. And these are all tested using diagnostic kits that are developed at CDC. We've most recently developed a multiplex real-time RT-PCR assay that detects both influenza A and B, as well as SARS-CoV-2 virus in a single tube. And that's really improved efficiencies within some of our laboratories around the US. But of those 200,000 specimens, about 6,000 of those go through our high throughput sequencing pipeline. We use NGS strategies to sequence directly from clinical specimens. And that's often the first sign of a human infection with the zoonotic virus, including H5 strains as well. Talk more about that as we move along. And then of those 6,000, about half of them end up in virus culture and are phenotypically characterized. The first step is to do antigenic characterization to look at cross-reactivity patterns with either human or animal antisera that's been developed against vaccines, or reference strains. And then we also have teams that are doing antiviral drug susceptibility testing to be sure that the recommended therapies that are available to treat all different types of subtypes of flu are also effective in preventing disease or treating someone infected with a pathogen. And then finally, a small number of those phenotypically tested viruses end up as candidate vaccine viruses, and this includes both seasonal as well as pre-pandemic candidate vaccine viruses, and I'll touch on those points as well. In addition to the domestic surveillance, the influenza division at CDC is also a WHO collaborating center and is part of a large global network called the Global Influenza Surveillance and Response System, or GISRIS. And the GISRIS network is coordinated through WHO, but is made up of more than 150 member states that include national influenza centers in those member states. And so this is a large network of laboratories. You can see in the map everywhere there's a triangle that represents a national influenza center that also does frontline testing of different surveillance platforms, primarily influenza-like illness and severe acute respiratory illness, in order to collect specimens from around the world. And then those samples are distributed among the 7 WHO collaborating centers that are shown with the circles on the map that are found in different regions of the world. In addition to the CCs, we also work with 12 WHO H5 reference laboratories that are also doing characterization of zoonotic viruses, but also doing quite a lot of surveillance in the animal hosts themselves to look for different animal influenza strains. Here at CDC, we also have some collaborations to do international field surveillance directly in animals. So I won't go through this entire list, but we coordinate with quite a few countries to also be able to do sample collections in animals at what we consider to be important animal-human interfaces. And this gives us a lot of data, again, early warning on emergence of new strains, new subtypes, as Isabella and Thomas have both mentioned, looking for different evolutionary traits, mutations in viruses, as well as reassortment patterns in those animal viruses. A lot of the surveillance that I've mentioned, as I said, is performed because we have standardized diagnostic assays that are used across these networks of laboratories, both in the US and among the WHO Global Influenza Surveillance and Response System. And so we are able to continue to develop and do performance evaluations of, again, the multiplex flu SARS-CoV-2 assays, but we also develop and distribute subtyping assays to detect seasonal H1 as well as seasonal H3s. We have influenza B lineage typing assays to detect and discriminate between B. vic and B. yamagata strains. And then for the zoonotics, we've also developed both an H5 assay as well as an H7 Eurasian lineage assay that allows us to detect potential human infections with both of these subtypes. And the distribution of these reagents as part of that frontline surveillance is coordinated through ATCC and a contract that the US government has with ATCC to establish what's known as the International Reagent Resource, or IRR. And as you can see from the map, everywhere there's bluish color that indicates countries that are receiving diagnostic test kits from IRR. And indicates just the huge number of countries that are able to take advantage of that resource in order to, again, to standardize some of the approaches for that frontline testing of influenza specimens that are coming in through various surveillance platforms. Another feature of the surveillance across the WHO network is also enhancements to sequencing, because of the SARS-CoV-2 COVID-19 pandemic, there's been a lot of capacity building across laboratories worldwide, and we work with those laboratories, not just at CDC, but again, across the network to improve and enhance the ability to do high throughput sequencing directly from clinical specimens so that we can identify changes in viruses, identify novel reassortment events. And in some cases, when the diagnostic tests fail or maybe inconclusive, we can back up that data with actual sequence information and confirm specifically what subtype or what lineage may have caused an infection. We also do the same to develop new strategies using different techniques. I think the one that most of us have been focused on recently for rapid sequencing is Oxford nanopore technologies. And so there's also been major efforts worldwide to enhance the ability of laboratories to do rapid sequencing using Oxford nanopore technologies and the various tools that are now available through ONT. We do this in the US, so we coordinate with some of our state public health laboratories that act as regional hubs to receive specimens from around the US, including Alaska and Hawaii, as well as Puerto Rico, to be able to receive samples and quickly sequence those. And then that data is fed back into internal databases that allow us to do real-time analysis of specimens that are received across the US. And then there's been also efforts, again, to do the same among many of the national influenza centers worldwide. And so this map shows countries that have been trained and are now receiving reagents to be able to do Oxford nanopore technology sequencing as part of the WHO effort to enhance genomic surveillance for respiratory pathogens at a global level. And so we continue to do training of these laboratories as well as to be sure that they're properly using the reagents in order to do full genome influenza sequencing as part of that effort. And of course, the important thing is how all of this data is then used for pandemic preparedness measures. And so we consider this a genotype to phenotype analysis to give us information as quickly as possible to do a number of things. It starts with identifying emerging lineages of new viruses, again, looking at mutations in viruses as they are identified to try and understand how those mutations might be impacting phenotype of the virus. But we also want to know how these strains are disseminating either at the country level or at the global level. And so we can do some mapping and do some proportion testing to be sure that we know where these viruses have been detected. Another big part of this is antigenic characterization, which I'll go into more detail. That allows us to make recommendations for which seasonal strains should be recommended for vaccines, but also which ones should be used for pre-pandemic candidate vaccine virus development. And then finally, there's a lot of efforts currently underway to think about ways to better integrate data, as Isabella mentioned, to be able to more rapidly share data across different networks, putting data into databases so that we can analyze it quickly and be able to use it, again, to make predictions about where we want to prioritize our medical countermeasures and ultimately how we might be able to even forecast or predict which strains might emerge as pandemic threats. Finally, this information is consolidated twice a year through the WHO vaccine composition meeting. And this is a meeting that happens in February and September every year to make not only seasonal influenza vaccine recommendations for both the northern and southern hemispheres of the world, but also to make recommendations on which of the zoonotic subtypes should be selected for pre-pandemic candidate vaccine virus development. I'm going to go into more detail on that. And so this is a really interesting group and a really, I think, attractive approach to being able to coalesce around a network that allows multiple laboratories from around the world, from both human and the animal health sector, to be able to talk about these things and to sit for a week, twice a year, share data, share thoughts, and ultimately make decisions about which of these strains need to be prioritized for pandemic preparedness efforts. So in the laboratory, this starts typically with antigenic characterization, and the hemagglutination inhibition test still represents one of the gold standard assays for being able to quickly characterize the antigenicity of a virus, again, relative to either antisera produced in animals or in humans that have been vaccinated with either seasonal vaccines or that may have participated in clinical trials with H5, H7, H9, and a few of the other zoonotic subtypes mentioned already. This antigenic testing can sometimes lead to a novel virus being identified that doesn't cross-react with an existing vaccine or reference strain. We put those viruses into ferrets to generate polyclonal antisera. That sera can then be shared among the network of laboratories that are doing the vaccine strain selection. So we're often shipping ferret anti-sera around the world so that other labs can benefit from that resource and reagent to do additional antigenic testing. When it's not easy to share viruses, we can at least share ferret anti-sera on a more regular basis. And that allows us then to do more clear risk assessment of these novel and more emerging strains. And so not only is this antigenic data, but we're also looking again at things like molecular markers from mammalian adaptation, receptor binding profiles, using things like glycan arrays to look at receptor binding specificity, antiviral susceptibility, and then ultimately the antigenicity compared to existing vaccines. There's a large number of other phenotypic tests that can be performed. I won't go into all of these for the sake of time. But again, this is a big part of the overall risk assessment that is also done both at the CDC and across the larger WHO network of laboratories. But moving quickly to the vaccine component of this. So we still focus primarily on development of vaccines that are egg-based vaccines. And so these can be generated using one of two different technologies. Classical reassortment is still used for viruses that grow well in embryonated chicken eggs. So you can mix two different viruses in the embryonated chicken egg and then select for the virus that has the H5N1 of interest in a background of the PR8, which is a high growth reassortant that allows the vaccine to grow well in eggs for manufacturing purposes. And then we also use reverse genetics to develop vaccines as well. This is, again, coordinated through a number of different laboratories. So the US CDC is one of several laboratories shown here in this list that contributes to the development of the seed strains that ultimately are used for both seasonal vaccine manufacturing as well as pre-pandemic candidate vaccine manufacturing. On the reverse genetic side of things have even improved to the point where we don't really require a virus in hand anymore. We can use synthetic approaches and use oligonucleotides to generate plasmids that carry the H5N1 surface protein genes that can then be transfected into cells with our PR8 internal genes. And then those viruses can be rescued and then put into embryonated chicken eggs. And we also now have cell-based vaccines that are manufactured by a couple of companies that allow us to use cells without having to use the embryonated chicken egg for passage or propagation of the manufactured vaccine. So the new technologies that are available through both synthetic gene approaches as well as more traditional reverse genetics using cloned RNA are really improving and enhancing the speed at which these vaccines can be produced. Now, not all of these pre-pandemic vaccines end up making it into the market, and so I'll go through briefly the timeline that's used to produce an influenza vaccine, thinking about these pre-pandemic CVVs. So it takes generally about 1 month to prepare the high growth reassortant, and then a couple of weeks to do the additional characterization and quality control to be sure that the CVVs that are developed meet certain criteria and pass certain safety testing. And that safety testing includes looking at whether or not the vaccine is attenuated in a ferret model to be sure that it's not causing any unexpected illness or severe disease or neurotropism in the ferret model. So a number of tests also go into performing safety and quality evaluation of the CVVs. Some of them, and this is a relatively small number, are selected for monovalent pilot lot production by manufacturers, and this is oftentimes coordinated, again, through WHO or other essential regulatory laboratories around the world that are able to fund the development of these pilot lots by manufacturers. And then some of those pilot lots will go into potency testing and preparation of reagents in the event that we may need to use them for a pandemic response. And so that time period takes much longer because the production cycle often depends on what's available within the manufacturing capacity of different vaccine manufacturers. So that might take a few months. And at the same time, we can also start clinical trials. And so a number of these candidate vaccine viruses have also gone into clinical trials so that we actually have data on the immunogenicity in humans that are participating in these trials. And then that gives us some sera generated from humans that allows us to do some additional testing, which I'll go into at the end of the talk. So finally, those that may end up being selected for actual vaccine production that may be stockpiled ends up taking about a 6-month period. So very similar to the time frame that it requires a manufacturer to produce a seasonal vaccine. It takes about that long also to produce these pre-pandemic candidate vaccine viruses. So we hope that we can at least get through the clinical trial for those that have been prioritized at the highest threat level in order to be sure that the stockpiles that are used both by the US and as well as a couple of other countries are prepared to have those vaccines available if there were a pandemic that occurred. So that's really important to consider, given that this can be a very prolonged process. And if we don't have things ready to go quickly, then these vaccines may not be very helpful for the first several months of a pandemic. So at least within the US government, we have created what we call the National Pre-Pandemic Influenza Vaccine Stockpile. There are currently multiple H5 avian influenza vaccines that are stored in the stockpile with about 20 million doses per antigenic variant. The same thing has also been done for H5N1 because of the threat of low-path H5N1 in China. Beginning in 2013, there were about 30 million doses that have been stockpiled for H7. And then in addition to H5 and H7, there's a number of other zoonotic viruses, including a few swine influenza viruses that have also gone through various stages of that vaccine production and have been prepared as stockpiled vaccines. Finally, there's also antiviral stockpiling to be sure that there's therapies available if there were a pandemic to occur. So then just to shift focus for a minute here on H5 in particular, you've seen some similar trees from Isabella already, so I won't go into too much detail other than to say that everywhere you really see these blue triangles, these represent clades of H5 viruses that have been circulating and have continued to circulate over the years. So many of them have gone extinct, and that's where they have become clear triangles, but others have persisted. And so for the majority of these clades that are shown, there have been candidate vaccine viruses produced, at least as those working seed stocks. And so there's a number, more than 40 now, of CVVs developed against H5 clades that provide antigenic coverage of those clades, and which could save us at least that 6 to 8 week time period in order to have a vaccine that could then go through the manufacturing process, as I had shown. To focus specifically on the clade 2344 viruses, again, you can see in red all of the different candidate vaccine viruses that have been developed against the various 2344 subclades detected in different parts of the world. And for the 2344Bs, there have now been 3 candidate vaccine viruses that represent antigenically distinct strains that are circulating in different parts of the world. And that includes a CVV, for example, that provides better coverage of those 2344Bs found in Africa versus, for example, the 2344Bs that are now circulating in the Americas. I'll skip over this slide because Thomas did a nice job of explaining the sporadic nature of human infection with these viruses. But to date, if you look at H5N1s beginning in 2021, there's been 19 human cases with really various disease outcomes, some that have been asymptomatic all the way through fatal infection. So it's still quite unclear to me exactly what is at the root of that difference, the range in clinical outcome of human infection with these viruses. And again, as Thomas mentioned, some of this may also be due to environmental contamination without actual infection of a human, if they've been working, for example, closely with poultry or are involved in a depopulation activity. So getting back to the vaccine development part, the US CDC receives samples through collaboration with the US Department of Agriculture, as well as after detection of a zoonotic case, and then coordination with some of our global partners. And we select which ones are likely to be antigenically different based on the amino acid profile or the genetic clustering profile of those viruses using traditional phylogenetic approaches. That allows us then to isolate viruses and to perform hemagglutination inhibition or virus neutralization tests. And then based on the reduction in cross-reactive titers relative to ferret antisera produced against existing CVVs, we may decide that we need a new CVV to cover a new antigenic variant. So for example, this is a data that was used for the February 2023 vaccine composition meeting that led to a recommendation for a new CVV targeting the American Widgeon South Carolina virus that was one of the first H5s detected in the Americas in 2021. So to date, there have been a number of 2344B candidate vaccine viruses developed. As I mentioned, there's 3 that have been selected. One has been developed both by CDC as well as the Food and Drug Administration in the US. But this is again in a background of many other candidate vaccine viruses that have now been produced against the larger 2344 clade. And again, these are products that have been developed by various WHO collaborating centers, as well as essential regulatory labs and a few manufacturers that have also participated in this exercise to develop these vaccine seed strains. Another thing that we of course do is antiviral characterization. Just for the sake of time, I'll mention that there's also this iterative approach to analyzing sequence data and then selecting based on the sequences which viruses should be phenotypically characterized to look at the ability of existing antivirals, including adamantane drugs, neuraminidase inhibitors, as well as PA inhibitors, to be able to effectively block infection with these viruses that have different mutations in the matrix gene, the neuraminidase or the PA. Some of these viruses then are further characterized using ferret transmission studies. We want to know whether or not these emerging strains may have different phenotypes in terms of their transmissibility. And so we can use both direct contact transmission models as well as respiratory droplet transmission models. And this is a work from one of our lab here that's run by Tarana Maines, showing the American Widgeon virus and the lack of ability to really be transmitted via direct or respiratory droplet transmission. But this is just one of now many genotypes that are found. And so there's a lot of other data now suggesting that some of these viruses do have the propensity to be able to transmit through direct contact, and then some that have shown some perhaps not efficient, but some respiratory droplet transmission in the ferret model. Finally, I'll just close by saying a lot of this data goes into finally risk assessment tools. So the CDC has developed what we call our influenza risk assessment tool or IRAT, that looks at all of these various elements, characteristics of viruses that allow us to prioritize these viruses for the studies that are done, and also to prioritize which ones should be selected for pre-pandemic candidate vaccine virus development or clinical trials. And so it gives us a metric to compare one virus to other viruses that have also been tested through the risk assessment tool. And so this just shows a scatter plot analysis of how viruses fall across this tool. And we're really looking at the likelihood that a given virus would emerge as a pandemic virus, and then if a virus did emerge, what the impact would be on the human population. The WHO also has a tool called the TIPRA that also focuses on some of the same elements that allow us to do that risk assessment, not just within the US government, but across the GUSRIS network. I'll close there. Of course, this is work across a lot of people, both in the US and internationally. I won't go through all the lists for the sake of time, and just say thank you for the ability to talk today. I appreciate it.
Thank you, Dr. Davis, because it's also an optimistic conclusion that there is preparedness. There are steps that have been pre-scheduled and pre-organized, and this is important and really hopeful. So before going to the questions, I just want to personally thank, apart from all of you who have been here and have been from the beginning till now and have asked all these questions, I want to personally thank 4 individuals who are with us. Matt Field, because he from the Bulletin of Atomic Scientists, because he has helped me a lot, and he has helped publicize the meeting. Helen Branswell, who is the prototype of the health journalist and knows very well avian influenza, has written about it for years. David Quammen, who is David Quammen, and we were very happy to have in Greece published his spillover book 2 years ago, and it remains an exciting book for the ones specializing on zoonosis. And Ioanna Souffleri, who is sort of the Greek Helen Branswell. So thank you for being here, and I hope it was worth it. I hope it was worth it for all of you. So let's start with the questions. And I will allow me to start with Helen Branswell, who is asking Tom Peacock, where he said that H5N1 might be a very possible candidate for a future pandemic, but then wouldn't decades of exposure to H5N2 viruses create some cross protection in the population?
Yeah, it's a very good point. So I guess this also applies to the Eurasian avian H1N1s that I mentioned as the swine viruses, because of course we have endemic H1s as well. So both the Chinese H3N8s and the Eurasian avian H1s, they are about almost as far away within the hemagglutinin trees as they can be while still being the same subtype. So although they are still H1s and still H3s, they are really quite distinct. It's thought for both those lineages, there's minimal cross-reactivity with viruses that have circulated in humans, at least for the last 50 years or so. So although it can, let's say, H1 protection against H1, as we know from 2009, that's not the case. An H1 can come along if it's antigenically distinct enough, it can still cause a pandemic. And that's the worry with the H3s, in that they're probably not cross-reactive enough with circulating H3s.
Okay, so allow me to forward 2 questions from Theodore Tsai first. Many of us know Theodore Tsai, and we are happy to have him with us. First, for Isabella Monne, could you expand on what's known on the infection of passeriforms? It would seem that infections of backyard birds and potential contamination of bird feeders and predation by house cats, there could be more direct spread to humans.
So passeriform luckily seems to be less susceptible than other avian species. There were some reporting, in particular in the most recent year, because this dimension with this virus has been, impressively increasing the number of susceptible species. But so far, as an example, sparrow seems to be less susceptible than the other. However, this does not exclude the role of these birds as vectors of the virus between the farms. So in a context of densely populated poultry area, the movements in the environmental contamination, even of passeriform, can absolutely play a major role. So the question is absolutely appropriate. Over.
Okay, so another question from Theodore Tsai about the recent recommendation by the EU Food Agency on vaccinated poultry. I think France is starting to implement it. How feasible is to vaccinate poultry? How long will it take?
This is a meaningful question for sure. Vaccination is a tool we do have and we have to consider, but this does not come without issues. First of all, economical, because you have to think about administrating thousands of animals these vaccines, and the vaccine we do have right now are expensive. In addition, there's the aspect related with trade. There are many countries that do not want to import, poultry products from countries which are using vaccines because of the trust behind this. And this is coming with the need to have an approach anytime you want to vaccinate, which is clearly aiming at the zero tolerance. This zero tolerance means a lot of testing, a lot of surveillance is needed in order to be sure that there's no circulation of the virus in the vaccinated flocks. So the cost to maintain vaccination is quite high for all these reasons, but still is a tool that absolutely in high-risk area for most susceptible species need absolutely to be considered. And there's some positive example about the use of vaccination, such as Hong Kong, where vaccination has been prevented in many cases, disasters differently than in other countries. So I think I can close here. I mean, it would be needed a very long time to describe all the challenges and the benefits from vaccination, but I stop here.
Now, two speakers have asked about whether climate change might have affected the emergence of these new strains.
Well, from an animal perspective, for sure, climate changes has been influencing the movements of hosts. Think about how important it is for all wild birds, temperature for breeding, availability of water resources. So it could be that there have been a different trend in the aggregation between different species and the places to stop over during their migration trips, but may have facilitated a different dynamic of interaction between different species. Then it's clearly different I mean, it's not so easy to estimate this kind of impact because there's the need to have more appropriate data that right now are not for sure completely available. But clearly we have to consider that the climate is influencing the movements of the main host of this disease, and for sure have an impact on the virus movements.
And now Michael Rampetti asks whether this aberrant H5N1 viral behavior may have existed for a while undetected and we just picked these trends. Did we go retrospectively back and search whether there was any previous circulation? Isabella, Thomas?
Well, I can be the first one, but I'm sure that also colleagues can add something on this. I do not think that this has been happening, despite this is the question I've been posing myself many times. Actually, I do remember when I joined the influenza division where I'm working right now, I mean, it was 2006, and I do remember that there was an impressive alarm caused by this Goose Guangdong lineage starting moving across the continents. And I do remember that at that time, we were receiving so many samples, but actually the peak in the number was similar, very similar to the one we had earlier this year when the mink story in Spain came out. So I do not think that we been not detecting properly the virus earlier. For sure, maybe genomic surveillance was less efficient, but still was already in place at that time. And I do remember whole genome data clearly were less available from wild birds, but from domestic poultry, the number of published sequences was acceptable, maybe less than now. But I do think that what we do have in a randomized solution, when we do all the analysis, we try to normalize the data in order to be comparable. And if you look at those data, the information about the impressive ability to resolve for this most recent virus is evident when you normalize the data. So I do think that it's not, there have been for sure gaps because back in 2006, we had a virus, clade 2.2, with mutation in the PB2 gene, the 627 mutation key, that was most probably acquired from an unknown mammalian host. So for sure we need something, but I would say that the situation we are currently facing in terms of animal perspective is, I mean, unprecedented.
Now, What is the overall the effect on the overall health of ecosystems, the potential consequences on wildlife conservation efforts?
Well, this is a really complex question to be addressed. I'm a veterinarian, not an ecologist, but I would say that we are not exactly in the right time to get to conclusion in this perspective. We do not know which is going to be the impact in the long term, because we do not know how the different species affected are going to recover. I mean, each species has a different, let's say, capacity to recover from mass mortality. Some species have a high reproductivity rate and they can replace progeny quite easily. Other, like long life animals, which invest on few progeny, on few chicks, they clearly having a later also sexual, they become mature quite late. It's clear that the situation for those species may be terrible. Which is the impact of losing species? Well, it's enormous, because these animals are maintaining an ecosystem that is much more complex than what we think. There's a lot of species that missing one of them, it means maybe becoming with an abnormal number, as an example of insects that may drive other diseases. We need to think about also the conservation perspective as an example for pinnipeds. I mean, many sea lions, elephant seals have been affected. What the impact of losing mammal species that are food for other species, such as, I mean, many cetaceans. It's a complex scenario, and in the long term, we do not know what is going to happen, but we need to monitor much closely, because it's not just the problem to think about a human pandemic. It's already a disaster, even if it stays as a panzootic event, because animals are part of our life, and we need absolutely to protect them. Over.
Now a provocative question. Thomas or Charles, do you think a mutation of the flu or viruses might be bioterrorism? Or is there a possibility to use it for bioterrorism? Now, when we did such a model for potential bioweapons back in 2009, flu was very low, but then we didn't even have coronaviruses in our list.
I mean, yeah, obviously, influenza's a virus that is in some ways would be well suited for that sort of thing, with the exception that, as with all respiratory pathogens, it's very difficult to target your enemies and not get a blowback from yourself as well, because as we know from 2020, these viruses do not respect borders, and they don't, you know. But of course, yeah, there's, I mean, since 2012, there's been a lot of talk about regulations and things, particularly in the context of H5, and you know, it's one of those things that the papers from 2012, the controversial papers, have actually been extremely useful for surveillance at the moment, trying to look for they are the roadmap for trying to understand what is going on in H5N1 at the moment, and whether we're seeing evidence of you know, mammal to mammal transmissibility. So it's a very complicated legacy, I think, those papers. Yeah, I think I've maybe skirted around, but slightly answered the question there.
Now, let me find the next question. Okay, do we have any new therapeutic weapons or possibilities for the future for severe cases of H5N1? But I think that this also applies to all influenza cases.
Yeah, I think that's right. I think, you know, perhaps one of the beauties of influenza A viruses is that there are conserved regions across the genome that exist between both human seasonal viruses as well as avian strains and swine viruses, et cetera. And so a lot of the therapies that exist currently work across the influenza A types, and those that are currently being examined, that are being considered for clinical utilization, also share the ability to work across subtypes. So I think there is some hope that there'll continue to be development of new therapies. Baloxavir is a very good example, of a recent antiviral drug that's now widely used in many countries.
Okay, so about the resource sharing and information exchange, I think you have answered this between countries. Do you have any prototypes of successful responses to H5N1 outbreaks apart from culling? Do you think that the containment of previous avian flu outbreaks was a perfect example of action, keeping it in the animal population or keeping it from expanding in the past? I don't know if the Hong Kong experience was a good one.
I mean, I can take this one maybe. The different highly pathogenic viruses that have been emerging, I mean, they were easier to be controlled because they did not involve wild birds. The problem with this virus is that you have a moving target to control. So with a high contamination as the one we have right now, we need to think about new strategies. And for this reason, I think also vaccination, that is something that in the past has been not used in so frequently, should be something to be considered. And now even Europe, which was a region, I mean, not that much into vaccination, there's now example of countries that are applying vaccination, such as France in particular, for ducks, which we know are really susceptible to the disease, and they are frequently clinically healthy. So reducing the spread in this way, we are really curious to see how it's going to, I mean, to answer this country after vaccination. So far, the measure have been always the same for this virus, biosecurity, culling, but clearly, as I was saying in my talk, we need to think about other solutions. And we need also to push a bit more research on vaccines, new vaccine that can be easily distributed. And that's the problem, as an example, for wild birds. You cannot vaccinate them because it's not that easy. There's the disturbance aspect that you cannot go there and take the animal to be vaccinated. There's no mechanism to distribute vaccines to these species which are living in the wild. And I mean, that's the need to work on this side, because there's, as I was saying earlier, the risk that some species are going to be, I mean, dramatically affected by this virus, and we need to find other way to control the disease, and not just pointing on biosecurity and culling of poultry species. Over. Yeah, for the other subtypes, and the other H5 and H7, which are not the descendant of the goose guangdong lineage, the control was most probably successful because they were a poultry issue. And so biosecurity, culling, early detection have been working efficiently. However, there are other highly pathogenic avian influenza viruses that are still causing problems in some countries, H7N9 in Asia, there's the Mexican H7N3, but I mean, these are still an issue of poultry, and you do not have a wild bird implication, luckily so far, but everything can change in a blink of an eye with this virus.
Now, the next question is for Tom, because I know he would like to answer it. How quickly is H5N1 adapting, comparing it to other viruses or influenza viruses that have transitioned to human hosts? And I would like to add how SARS-2 is still going.
Yeah, so it's quite difficult to say because although there are some lab studies looking at how quickly things can adapt, most of the evidence of what we have is quite, if we find a mammal infected with these viruses or a human infected with these viruses, how quickly they say switch their polymerase to E627K or whatever mutation. These viruses, I mean, in fact, there actually seems to be differences in the genotypes. We actually have some data to suggest that the viruses that were most similar to the ones in cats in Poland that we had in the UK in the last winter, I think, or maybe the winter before, I'm losing track of the year, they seem to, every time they jumped into a mammal, they would get E627K. But what's interesting is the BB genotype, which Isabella was talking about as the gull, the one that was very prevalent in seabirds rather than waterfowl, and that seemed to sustain over the summer. Generally, that didn't seem to get so many mammalian adaptations when it was jumping into, say, foxes. Although there is exceptions in the mink farms in Finland, because that is the genotype that caused that. So even within the H5s that are circulating in Europe, there seems to be quite a lot of variation. So it's quite difficult to kind of make any wide generalizations without going down to an individual strain level, I think.
Two more questions for you. It has been reported that in H5N1, the gain of MXA escape impacts its nuclear import. In one slide, you showed some strains that were resistant. Do we know whether these also have this defect in nuclear import?
So we haven't looked at that. We're very interested in looking at why these viruses, it's not unprecedented for circulating avian influenza viruses to be resistant, but it's not that usual. And we're very interested to see what's causing that maintenance and if there's something driving that or it's just chance. That might be something we should look at. Thank you for the comment.
I want to thank Dr. Davis because he had to leave, so he won't be available. We will try to answer any relevant questions. One more for Tom. What about adaptive mutations in PA or PB1?
So interestingly, the African H5N1s that had reassorted with the endemic H9N2s that Isabella was talking about earlier, we did do some testing on some of that, and interestingly, there's something about the PA from the H9N2s that we've actually described before, but we didn't realize that H9N2 also had that thing. But it seems to give the virus some sort of intrinsic ability to replicate in human cells, which is a little bit concerning. Without any, that's not mammalian adaptation. That's just something that's circulating that naturally has that property. So again, yeah, it's interesting. These viruses sometimes throw up interesting little things like that.
A question from Lucas Gonzalez on non-pharmaceutical interventions on protection. The clean air and masks, etc. These are lessons that we do have to remember from the current pandemic anyway. So yes, obviously. Now, yes, that we do have to build resilience on non-pharmaceutical interventions too. And okay, we have answered this. About the data on increased adaptations to human host over time, it has been covered. This is the same question again. About non-pharmaceutical interventions, Lucas Gonzalez is asking again. Yes, I do agree that we do have to clean the indoor air. This is indeed a public health revolution would be. There's a comment from Theodore Chai that West Nile virus was a mass mortality event for crows when it went in the US, but they were falling out of the trees, but the population has recovered. That's good. And I think that the final question I would like to answer, this is from Michael Rampetti. The current pandemic multiplied the anti-science and anti-vaccine sentiments. And science communication is in crisis. I do agree with him. he's asking if it is in crisis. I do agree with him, and that is why we were trying to put this together, because we have to make science not only understood but also popular and accepted, with all its shortfalls, with all its questions, with all its the methodology of science and the respect for science, not necessarily for scientists. I hope we did something with this, and I do think that we have done some good things here, and I hope that this webinar will echo through the ones who were present in the future and will be visible in the future. So I want once more to thank the International Society of Antimicrobial Chemotherapy, Phil Johnson. I want to thank Phil Johnson, who put this all together. We all want to thank Phil Johnston. And in the chat, there are the links of the Antimicrobial Society and its presence in social media everywhere. I want once more to thank Thomas Peacock and Isabella Monne. It has been an extreme honor to have you here. And I want to thank everyone else. So goodbye.